Biology, chapter 16, theory video 2. We are going to deal with control of gene expression. So now earlier we learned how the phenotype of LE gene, how it is expressed in relation to stem elongation. So now we are going to deal with how exactly it is done. We say that a gene is expressed when it is transcribed to a mRNA, and then this mRNA will be translated to produce the protein. So the protein, the production of the protein will give us the give out the expression that is needed. That is, if the stem elongation is going to happen, the stem elongation happens as a result of the production of the protein. um, then we need to understand that not all the DNA, not all the genes would be expressed at the same time. And also not done in every part of the body. For example, the gene for melanin production is not done inside your heart muscles. It is not done in any part of the inside cells. Melanin is expressed only in the skin cells. So in a prokaryote organism, you know what prokaryotes and eukaryotes are. In a prokaryote organism, we are going to deal with how the gene control and expression is taking place. Now, bacterium, E. coli, we will first deal with that and then move on to the eukaryotes. Now, for a long period of time, especially since the production of cheese, wine, and the different fruit products that required the involvement of a bacterium, people have been interested in bacteria, and as a result, we have been, for a long period of time, we have been studying about this. Now, B-galactosidase, it is also known as beta-galactosidase, which is also known as lactase or B-galactosidase. So this is all referring to the beta-galactosidase. This is used by some of the bacteria to hydrolyze lactose to glucose and galactose for their energy supply. So they need energy. So they need to, they don't have a developed system to digest whatever the materials that they take in. So as a result, what they do is they release the enzyme beta-galactosidase to hydrolyze lactose into glucose and galactose. And that can be absorbed to provide for their energy requirement. So this beta-galactosidase is an example for a structural gene, meaning that it codes for the production of a protein that is used by the cell. If the protein is used by the cell, the gene that codes for it is known as a structural gene. Then there are some structural genes. They are actually, they make part of the structure itself, right? So it doesn't have to be, the structural genes not only code for proteins that make part of the structure, but also if it is used by the cell, it is also a structural gene. So they produce some structures, protein structures, then at the same time many other structural genes have other functions, that is to produce enzymes or coding for enzymes. Then the expression of the gene is controlled by other genes which are around this, around this circular DNA molecule. They are known as regulatory genes. So the gene that codes for the production of a protein that is used in the cell is known as a structural gene. The uh, genes that control or expresses expresses that particular gene is controlled by some other genes. They are known as regulatory genes, right? So the keywords here that you need to remember: B-galactosidase, an enzyme that catalyzes the hydrolysis of lactose to glucose and galactose. Structural gene, a gene that codes for a protein that has a function within the cell. It could be a structural function or otherwise, but it is going to be known as a structural gene. Then a regulatory gene, then a gene that codes for a protein that helps to control the expression of other genes. So it helps to control the presentation or the function of the other genes. These genes which have structural and regulatory functions, they are found close to one another in a group, in a cluster. This cluster would be known as an operon. It is a functional unit of transcription. That is a functional unit of something that does the work, right? The operon is this cluster of genes which have a structural and regulatory genes. The operon that is responsible for the production of lactase in bacteria is called the lac operon. So it is the group of these genes, right? The lac operon, the structure is shown in 16.13. So this is the lac operon gene structure, right? So if you look at this carefully, you will be able to understand what it is. There is more detail here. In the bacterium E. coli, the number of molecules of beta-galactosidase present in a cell varies according to the concentration of the lactose in the medium in which the bacterium is growing. So for E. coli, the number of beta-galactosidase that is going to be present is going to depend on the amount of lactose present in the medium it is growing. Because it is actually, there is a mechanism that, the external environment will actually decide what is going on inside. The quantity of the enzyme that is produced is altered by turning this beta-galactosidase gene on or off. It doesn't function all the time. It is not expressed all the time. So then that is done by the regulatory genes. So that is present inside the lac operon. Then there are other structural genes in this lac operon which have a function other than coding for beta-galactosidase. There is LAZ coding for B-galactosidase. Then LAC-Y coding for permease that allows the lactose to enter the cell. It makes it permeable, which allows lactose to enter the cell. Then LAC-A coding for transaxilase, that is responsible for the transcription. So transcription of all these genes is controlled by the same promoter. So these are words that you need to understand and remember as names. So there is LAC-Z, LAC-Y, LAC-A. The lac Z codes for beta galactosidase, then lac Y code for permease, then lac K transaxytilase, and I'm not really sure about the pronunciation, but that is those are the functions of these things. And these three structural genes are responsible for each function. Then there is a part of the bacterium DNA which is known as a promoter. That part promotes or controlled by this particular promoter, and they are transcribed all at the same time. Then this is what happens if there is no lactose in the medium where the bacterium is growing. Then there is no need for the production of beta galactosidase because there is no lactose. Then what happens is the regulatory gene would code for a protein called a repressor. Then the repressor binds to the operator region close to the gene for beta galactosidase. Because there is a repressor which is attached to the operator, the RNA polymerase cannot bind to DNA, and as a result, no transcription of the three structural genes would take place. You can remember in earlier parts you must have learned about transcription. So that is not possible because this is like the inhibition of enzymes. The same thing happens here. The repressor protein has two binding sites. They can bind to DNA at one site and to lactose from the other side. When lactose is bound to the site, what happens is the shape of the repressor protein changes to allow the DNA binding site to close. So here you can understand this through this diagram. There is a so this entire thing is known as the apologies. This entire part is responsible for the coding of the protein. So that is the DNA molecule. So in this DNA molecule you have the promoter for regulatory gene, then regulatory gene, promoter for structural genes, operator, then the structural genes lac Z, Y and A. So they are all they are in its function. Then what happens is there is a oppressor protein here. This oppressor protein when RNA polymerase cannot reach get attached to the DNA, as a result it will not beta galactosidase will not be produced. If lactose is present, then what happens is it binds to LAC repressor protein. So it attaches itself to the repressor protein. Lactose will attach to the repressor protein. Then that results in the detachment from the DNA. That allows the RNA polymerase to bind and transcribe the operon structural genes. So then as a result you will get beta galactosidase, and as a result the breaking down of lactose would be a possibility from there onwards. So these are the steps that happens when lactose is present in the medium. Lactose will be taken up by the bacterium, and lactose will bind with the repressor protein, and that distorts the shape, and as a result DNA, it cannot, it can no longer, it will, the shape is distorted because they are unique. They cannot attach to the operator site, and as a result, it will, uh, it will be removed. Then mRNA will be produced at the three structural genes, and which results in the production of the protein. So this results in the switching on of the gene. Then what happens is lactose breaking down can continue. So this mechanism allows us to understand that beta galactosidase, permease, and transactylase is produced only when there is lactose. The bacterium should live in a medium where lactose is present. So this actually this results in this mechanism, this kind of a negative feedback mechanism allows the bacterium to save energy because it is not doing unwanted controls within the body. So whenever the sugar is available, it can be hydrolyzed because the process can begin once again when there is lactose. This enzyme, beta galactosidase, is known as an inducible enzyme, meaning that it is produced or synthesized when only its substrate is present. Inducible enzyme, you know what is meant by inducing. So the presence of a substrate induces or causes the transcription of the gene for the enzyme. So the oppressors would be removed off, and as a result, the transcription can continue. The effector molecule. So the effector molecule in this particular scenario is lactose. So that the effector molecule binds with the repressor, but distorting its structure, and as a result, it will not be able to bind to an operator, and it is going to proceed. Then there is a other enzymes, whatever the other enzymes, which are known as repressible enzymes, would be controlled in a different way. Here what happens is the effector molecules attach to the repressor. As a result, it can attach to the operator. That stops the transcription. That is how the repressible enzymes work. They are entirely the different, it is stopped by the attachment of the effector molecules. I hope you understood. Let's write down a note, and after that, we'll come back to eukaryotic cells. Write down. A gene is expressed when it is transcribed to mRNA, and mRNA translates to produce a protein. A gene is not, leave a line and write, a gene is not expressed in the same manner everywhere or at all times. Example, Gene for melanin production will never be expressed in heart muscles. Will never be expressed in heart muscles. Write down gene control in prokaryotes. In E. coli bacterium, the gene that codes for the production of the enzyme, beta galactosidase, within brackets also known as lactase, is considered. Leave a line, put a star and write down. Beta galactosidase is used by bacteria to hydrolyze lactose to glucose and galactose, which can be absorbed by the bacterium. Glucose and galactose, which can be absorbed by the bacterium. Leave a line, put a star and write down structural genes. These are the genes that code for the production of a protein that is used by the cell. Within brackets, having a structural function or any other function in the body, in the cell. Having a structural function or any other function in the cell. Next point, regulatory genes. These are genes that codes for proteins that help to control the expression of other genes. Leave a line and write. These structural and regulatory genes work together as clusters and are known as operon. Leave a line and write. The operon for the production of lactase in bacteria is lac operon, LAC operon. In Escherichia, that is E. coli, in E. coli, the production of beta galactosidase is decided by the presence of lactose in the medium in which the bacterium grows. Presence of lactose in the medium in which the bacterium grows. If there is no if there is no lactose, gene is switched off and switched back on when lactose is present. The lactoperone, leave a line and write lactoperone, there are three structural genes, LAC-Z, I put a dash and write, coding for beta galactosidase, then LAC-Y, LACY, coding for permease, which allows lactose to enter the cell, or which makes the cell permeable to lactose. Then LAC-A, coding for transacetylase, transacetylase, that is how you pronounce it, transacetylase. That is LAC-A. Leave a line and write, the transcription of all three genes is controlled by the same promoter and is transcribed at the same time. When there is no lactose, leave a line and write, when there is no lactose, regulatory gene codes for a protein called repressor, which attaches to the operator region preventing RNA polymerase binding to DNA. This results in no transcription taking place. Leave a line and write, when lactose is present, lactose will bind to repressor molecules, distorting it. This removes the repressor, allowing transcription to take place. Pause the video and draw this figure 16.13. That will give you a better idea as to how it is done. Enzyme beta galactosidase is an inducible enzyme, underline inducible enzyme, which means that it is synthesized only when its substrate is present. Leave a line and write. The other types of enzymes called repressible enzymes are produced in a different way to inducible enzymes. Here binding of the effector molecules will make the repressor to bind to the operator, which stops the transcription. This is the exact opposite of what happens in inducible enzymes. I hope you understood. We will go to the next one in the next video. If you have any questions in this, please ask.